Well, let's continue. Well, remember in the last slide I give you a kind of a demonstration about how double helix is formed by using my two arms. So yes, remember the bases on each strand, they are connected with hydrogen bond. Okay, they are connected by hydrogen bond. That's why hydrogen bond is the most important bond in biology, not James Bond. And then you also need to memorize the complementary base pairing, this rule. A pair with T, and then G pair with C. So how do you memorize this? Let me give you a tips. So you have A, T, G, C. Okay. A and G, remember from previous slides, we call it purine. And T and C, we call that what? Pyrimidine. Pyrimidine. Pyrimidines, okay? And according to the complementary base pairing rule, A pair with T and they form 2 hydrogen bonds. And G pairs with C and they form 3 hydrogen bonds. So remember on our campus, okay, well, for West LA College, we have a building called General Classroom, okay? It's right across the cafeteria. A lot of English classes, a lot of classes in the General Classroom building. So in here, how you can memorize that this is ATGC, meaning that at GC building or at General Classroom. So that helps you to memorize it, and also you can think about it as at guidance center. At guidance center. Well, but for us, if you go to graduate school, we will teach you this. Instead of going to at General Classroom at GC building or at guidance center, if you go to graduate school, we'll teach you at Gentleman's Club. Okay, that you will never forget. All right, the two strands are anti-parallel. This is also an important point. Anti-parallel, meaning that they are in opposite directions. Right now, it is hard to explain to you what does it mean. 3', 5', 5'N, 3'N. In the next few slides, we will show you the direction of the sugar phosphate backbone depends on the 3' and 5'N on the sugar. So the 3', 5' basically is the numbering of the carbon atom on the pentose sugar. So we put number 1, 2, 3, 4, 5 to the 5 carbon in the pentose sugar. And then the 3' carbon and the 5' carbon, they are connected to different group, and then it forms a chain in the backbone. And because of that, one strand of DNA is a 3' to 5' direction, and then the other chain of the DNA strand is a 5' to 3' direction. So they are in the opposite direction. That's why we call it anti-parallel. Don't worry if you don't understand what I'm talking about now, because I'll show you a picture to tell you what it means by anti-parallel. But right now, just memorize ATGC, okay? So this summarizes everything, but they are very small. So in the next few slides, we will show you individual picture, the magnifying one. But as you know that, okay, DNA is a double helix structure. You can see that this is one strand and this is another strand. So the part that I draw the black line next to it, those are the sugar phosphate backbone. So in the picture, it is like the blue thin ribbon. The blue thin ribbon, they are the sugar phosphate backbone. But you see that this part, the colorful stairs, so it's like a ladder, twisted, it's like a twisted ladder. The colorful steps or colorful staircase, those are the bases, and they are connected by hydrogen bond. When we untwist the DNA double helix, we will get a ladder structure. Now it is very clear. G form 3 hydrogen bond with C, and T form 2 hydrogen bond with A. Okay, and then you can see that they are called anti-parallel because you can see that from 5' to 3', and then the other one is 3' to 5'. So they are in the opposite direction. But what do we mean by 5', 3'? Okay, I already mentioned that it is the numbering for the carbon atom in the pentose sugar. Let's take a look. So as you can see in here, the carbon atom start with number 1 or 1' is the one that is connected to the nitrogenous base, and then it just continue. And then it just continue numbering the carbon in the so-called clockwise direction. And then the 3' carbon, what's so special about the 3' carbon? The 3' carbon is connected to a hydroxyl group, OH. And the 5' carbon, what is so special? The 5' carbon is not does not form the pentagonal structure of the sugar. It is separated from the pentagon, and it is connected to the phosphate group. So it is connected. So 5' connected to the phosphate group, 3' connected to the hydroxyl group. So let's take a look. Well then, so again, which molecule have you learned from this class? The most similar structure is DNA, the polymer of DNA. Well, think about this. I'll give you some time to think about this. So DNA replication is basically like making a Xerox copy. So it is copying one DNA into 2 identical double helixes. Where do you see DNA replication occur? Remember what we covered a few lectures ago? Mitosis. In mitosis, during interphase, the S phase, the synthesis phase, is where DNA replication occurs. And remember, in mitosis, it is one mother cell become 2 daughter cell. So it is very, very similar to DNA replication. DNA replication is also one DNA becomes two identical DNA. The mother DNA becomes two daughters DNA. And the two daughter DNA, they are identical to each other. And they are also identical to the mother DNA. So it is equivalent to mitosis. But mitosis is the replication of cell. DNA replication is the replication of DNA. So DNA replication is, you can assume that it is the mitosis of DNA. Okay? And as a matter of fact, before mitosis occur, we need to have DNA replication to occur in S phase. So DNA polymerase is one of the 3 enzymes that I want you to memorize. DNA polymerase. So what is DNA polymerase? If you remember, in the previous lecture, I mentioned that, okay, ASE, whenever you see a word with a suffix ASE, that means it is an enzyme. What does this enzyme do? This enzyme make polymer. Okay? What kind of polymer? It makes DNA polymer. This enzyme makes DNA polymer. So that is the function. Basically, the name of the enzyme tells you the function. We learned in the previous slides the anti-parallel, what it means. So now we introduce another keyword, semi-conservative replication. What do we mean by semi-conservative? Semi-conservative meaning that, so alright, this is my poor drawing. This is a mother DNA. Mother DNA. It's difficult to draw. I'm sorry. I'm not a good artist. Okay, I'm not impressive. I'm not that good at drawing. Alright, okay. These are the two daughter DNA. Two daughter DNAs, okay? The reason I use two different colors to draw the daughter DNAs because they use the original strand as the template and then to produce a new complementary strand. So the black one is the original mother strand and the green one is the new complementary strand, meaning that the green one, they are newly made. The black one is the original, the old one. That's why we call it semi-conservative, because the new daughter cell, half of the strand, or not half, one of the strand one of the strands is from the original. That's why it's semi-conservative. It conserves the original one. So it says that one new strand, and then one parent strand, one old strand, one parent strand. Okay, one new strand, one parent strand. That's why we call it semi-conservative. Yes, they are identical to the parent molecule, and they are also identical to each other. I already told you that the two daughter DNA molecules, they are identical to each other, and they are also identical to the original mother strand. So this is the summary of the DNA replication.